Welcome to Ask a Scientist. I'm Lynn Philman, independent artist and author for genomic science and co-chair of this session. It's one of many during this 3-day scientific conference organized by the Genome Writers Guild and the Mayo Clinic. Tonight's event is special. It's designed with the public in mind. Easy on jargon, yet plenty of fascinating research to captivate all you scientists out there and the rest of us.
And I'm Adam Challa. I'm a graduate student at the University of Minnesota and the co-chair of this session along with Lynn. And we're here with two amazing scientists and science communicators. Our keynote, Dr. Alison Van Eenennaam and Dr. Anil, Challa. And we'll have a Q&A session following their talks starting at about 7:45.
Okay. Thanks, Adam. Before formally introducing Anil and Alison, we have some housekeeping tasks, and we'd like to tell you about the Guild and why Adam and I are members. The Guild is a nonprofit genome engineering society. Our mission is to build a better future for humanity through genome engineering and public education. We're unique in the world of scientific conferences and organizations. Our members are scientists, educators, entrepreneurs, people in government, writers, artists, science communicators. We welcome anyone supporting the responsible use of the powerful genome engineering and editing tools. It's science for good.
And promoting responsible genome engineering, it's a joint effort. So here at the Genome Writers Guild, we aim to facilitate this effort. We do this by focusing on ethics, science, and public communication. The broad spectrum approach is exactly what got me involved in this group in the first place. And whichever point seems to fit your interest best, or if you feel that there's other places that we should really focus, we really could use your voice.
Yeah, we can. And so Adam, just a little bit about you. You're vice president of the Guild and an early career scientist. So tell us what you're up to.
So my main work focuses on using genome engineering technologies in order to control pest insect species such as mosquitoes. So the goal is ultimately to help prevent diseases like malaria. And Lynn, so you're an artist. So how is it as an artist that you got interested in genetics of all things?
Yeah, well, I got hooked way back in the early stages of sequencing ancient DNA, when genes and bones were finally put together to understand human evolution. When DNA test kits mapped migrations of our and my distant ancestors, and now sequencing the whole genome and maybe fixing it if I get sick. I mean, who wouldn't want to be part of this? So I started to write and draw about our genome stories to communicate the value and wonder and to push back on the anti-science and anti-GMO forces. After all, genome engineering impacts our daily lives. It's why scientists and the rest of us need to talk to each other. Why art, poetry, music, dance, journalism, all the cultural variation is needed to show the beauty and value. And like you said, Adam, we need all our voices to bring us together. Okay, just a few more details and then on to our speakers.
Yeah, so just as a big overview, this conference is a safe and inclusive space, so any derogatory, abusive, or harassing behavior will not be tolerated and is grounds for immediate expulsion from this meeting. We do expect all our participants and presenters to uphold a high ethical standard. So we will be taking questions at the end of when both talks conclude. So there's a Q&A window at the bottom of the Zoom call, and you can use that to type in your questions. And if you are attending the full conference, there'll also be a discussion board and a breakout session afterwards. In the Q&A window, you can just type in your session, and you can also upvote questions that that you would like answered. And so with that, without further ado, I'd like to introduce Dr. Anil Challa. Dr. Challa is an adjunct professor at the University of Alabama, Birmingham. He received his PhD in molecular genetics from The Ohio State University. And his laboratory focuses on understanding the mechanisms underlying cell movements during vertebrate embryo formation, mainly using zebrafish as the model system. With CRISPR becoming such a large part of biological study and the world in general, Dr. Challa recognized that undergraduate biology educators require effective classroom materials and pedagogical methods. So based on a series of course-based undergraduate research experiences at UAB, he began teaching workshops on the topic. Today, Dr. Challa will be sharing how these workshops came to be and how they impacted CRISPR education. Dr. Challa?
Okay, so the title of my talk is CRISPR in the College Classrooms. And the story begins when I was in college. So not very different from maybe many students. A lot of things were happening in college, but my imagination was captured by some of these images of science, mainly because of the excitement of discovery. And I think this has probably not escaped anybody's imagination in terms of the discovery of the structure of DNA and as a molecule of life. In addition to the excitement of discovery, there is, for the youthful me, it was very exciting to find out about the race to be the first in discovering some of these things, and the ability to actually make new tools and products using some of these technologies in genetics. For example, one of the things, the biggest thing that captured my attention was how recombinant DNA can turn bacterium into hormone-making factories. And one thing I'm going to plug here is that plasmids are treated with restriction enzymes, which make them open up. You can do all these kinds of really fun and exciting manipulations of DNA, and that really was very attractive to me. And it wasn't, you couldn't escape the fact that all of these things were actually having an ethical, legal, and social impact. And this was the background in which I got excited. And to set the stage, this is a key paper which was talked about by Rich Roberts in 2005. So in December 1971, a key paper appeared in the Proceedings of the National Academy of Sciences that set the stage for much of what is now routine. In that paper, they showed for the first time that the restriction enzyme could be used to produce specific fragments of DNA. And this was, while it might look very routine right now, that was extremely exciting to be able to see these pieces of DNA on a gel. So that being the background, that in fact catapulted my research career, and I went to different places, and I ended up at the University of Alabama at Birmingham. But before I moved there, I actually learned about genome editing a little bit by a short visit to Minnesota to Dr. Dan Whitehouse's lab in 2011 to learn about the TALENT technology for in vivo gene manipulation. And soon after, as I said, I moved on to assume a position in the transgenic and genetically engineered models core facility there to work with zebrafish and also to start working with mice and rats. So this is a time when CRISPR got everywhere, and almost like prophetic, like the 1953 Watson and Crick paper, there is this one sentence in the 2012 paper which says, "Our study reveals a family of endonucleases that use dual RNAs for site-specific DNA cleavage and highlights the potential to exploit the system for RNA programmable genome editing." And this was exciting, and and the rest is actually history. And at UAB, while I was having a day job of working with all these genome editing technologies to make animal models, I had an opportunity for participating or contributing to the honors program. So there was a freshman course in the second semester where students would experience research methods. So they would participate in a lab experience either in many different disciplines, but molecular genetics being one of them. So around the same time, I was heavily influenced by this document called Vision and Change for undergraduate biology education, which basically called for action. And the 3 points I'm going to emphasize here is that it said that we need to engage biology community in the implementation of change by ensuring that all undergraduate students have authentic opportunities to experience the processes, nature, and the limits of science. They also said that we have to encourage its application when developing courses and to create active learning environments, mainly for the first year biology students. So with these 3 action items in my mind, and with the focus on changing the interest from experimental techniques to the process of science, so that techniques as a means and not as an end in themselves, I created a course-based undergraduate research experience, essentially because it involves whole classes instead of single students who are already motivated. So this addresses issues that are there with regards to making inclusive environments for research opportunities for undergraduate students. So the CRISPR, we called it the Cure with CRISPR, where students dabble with a little bit of bioinformatics and a little bit of laboratory work and experience the whole fun and excitement of gene editing. So, I mean, I will not belabor the whole details, but students actually start with, these are freshman students in their 2nd semester. They start playing with DNA models and eventually end up doing laboratory work, eventually presenting their authentic research to a university audience. So I encourage you, I mean, I can post these links. You can actually see what students kind of go through and experience. So why freshman cures? And what was exciting to me was to hear students say that, it actually builds a broader and a complete context. Here's a quote from a student which says, later courses seemed easier because we had done things with our own hands. And it actually primed students. It said that, some students said, it was influential, or this cure was influential in my having the confidence to join a research lab. And it was not always that students got excited and they said, oh, I want to become a researcher. But early awareness, where some students felt that while I enjoyed the course, I realized I did not want to do this kind of work in the long term. So both, in a positive way to push them towards research, and in an alternative way where they actually realized that they enjoyed the experience, but they actually wanted to do something else. So then the icing on the cake was we were actually over 3 years, and the course continues to be offered, was a research publication with 16 freshman undergraduate students, 2 senior undergraduate students who were actually working with me on their senior thesis project, a graduate student who helped me finish this work, and myself. So with all this excitement, we felt, this probably should be happening in many colleges and many undergraduate classrooms. It turned out that I was at a ASCB meeting, and I found out that with a colleague that this seemed to be an interesting platform to introduce other undergraduate teachers. So that was the emergence of a national teachers workshops from Cure with the CRISPR into a class, to make it a classroom experience broadly. So my partners in crime were Dr. Mike Wolniak, who was the colleague I was talking about at ASCB, and a conversation started off, and his student, Luke Bloodworth, ended up in my lab working in a summer project. So both of these, a chance meeting with Mike and a deliberate plan to work with Luke over the summer, ended up with formulating a program where we wanted to get teachers involved. And the first instance was a 2017 major workshop at the Association for Biology Laboratory Educators at the University of Wisconsin-Madison, where it got a huge, we actually found out there's a huge demand for the CRISPR modules from the teachers. So fortunately, with support from NSF, we were able to organize a hands-on workshop in 2018 at Ohio State University, which then moved on to University of Minnesota in 2019. The essence of these CRISPR workshops was to connect syllabus to CRISPR technology, where some fundamental ideas and flow of genetic information can be nicely connected to the CRISPR workflow. So, as I said earlier, we actually introduced teachers to genome browsing and CRISPR analysis using bioinformatics tools. And then we basically created modules which can be done in a very simple way, either in a short form or in a semester-long form, where students can experience a bunch of really exciting things with this technology. again, I'm not going to spend a lot of time on the details. I'll be more than happy to talk about it in the breakout sessions. And we've actually been able to communicate this excitement of the teacher workshops to the broader audience. And this sums it up for us. There are several elements that we, in this particular feedback, I got from one of the workshop participants. And she said, I had my students replicate what we did in the workshop with their genes of interest over the course of the semester, and each of the groups had multiple guide RNAs that worked to cut their targets in vitro. So that's the mainstay of this whole teacher workshop. The students did a great job and were excited to feel like they were doing cutting edge science. I relied heavily on the tools shared in the workshop, and I would not have been able to do it without you all. So that's really a very nice feeling that we were able to help teachers. I think that teaching this capstone gave me a leg up in the job market, and I just accepted a tenure track position. So that's always a great thing. And she started off last fall. So she thanks for the workshop, and she hopes that we will be able to continue to offer this workshop. So with that, I think my time is up a little bit. And if there is time, I'll be happy to take questions. So I'll end this with just to, we are in the COVID times. We have to acknowledge that. So the excitement could not stop us, even if it was the COVID. And as we speak, we are offering a virtual CRISPR in the classroom workshop using Zoom. And this is a snapshot from Wednesday. That is actually, actually yesterday. And we are having a lot of fun in this teachers workshop as well. With that, I'll actually stop here. I'll thank a few people who are very important. Dr. Diane Tucker, who was instrumental in me getting involved with the SciTech Honors program, and helped me actually create this CRISPR cure at UAB for the freshman honor students. And these are all the partners in crime for the teachers workshops. And I would like to acknowledge my students, Sammy and Sarah, who kind of were adventurous and did their theses with me. Luke, who did a really interesting work in the summer of 2016. Ashley, who was a graduate student then, postdoc later, and now a new faculty member at Jeff State. And Jay Bhatt, who was a graduate student then and now a faculty member at Creighton. With that, and finally, I'd really like to thank NSF, because it gave us the support to take this nationwide. And thanks for your attention. I'll take questions from now or later. Thank you very much.
CRISPR critters and talent tofu. The title of Dr. Alison Van Eenennaam's talk is already making me smile. And curious, what's it all about? It's an extraordinary gift to engage us, spark our curiosity, and that's what Alison does with TED-style talks, at the Dr. Oz Show, on NPR Science Friday, and her appearance in the documentary film Food Evolution. Her talent as a sci communicator is grounded in scientific work as the cooperative extension specialist in the field of animal genomics and biotechnology at the University of California on the Davis campus. She received a Bachelor of Agricultural Science from the University of Melbourne in Australia, and an MS and a PhD in both genetics and animal science from UC Davis, where her current research and outreach program focuses on livestock production systems like the development of genome editing approaches for cattle. She's the bovine genome coordinator for the USDA National Animal Genome Research Program. Among her many awards is one reserved for scientists that excel in public outreach. She's an elected fellow of the American Association for the Advancement of Science. I had a sneak peek at her talk, and you'll see how she weaves a deep affection and care for animals since childhood in context with science discovery and gene editing basics, making us feel smart, able to understand how biotech makes livestock healthier and plants more nutritious with less chemicals for a sustainable environment. So now, let's watch Alison's talk and jot down your questions.
Good evening. My name is Alison Van Eenennaam, and I'm a geneticist at the University of California in Davis. Although clearly, as you can tell by my accent, I did not grow up here. I was born in Australia, 10 years after the famous 1953 paper reporting the double helix structure of DNA, enabled by a rather famous photograph out of the lab of Rosalind Franklin, and about 10 years before the first recombinant DNA molecule was produced. Growing up in 1960s Australia, I was always surrounded by weird and wild animals, and summers spent on my auntie's farm riding horses, working cattle, and being chased around by various members of Australia's venomous army, including an echidna mother who loudly chased the pony I was riding in an effort to protect her puggle, the terminology for a baby echidna, are my fond and enduring childhood memories. I also clearly remember frequent news reports of the tragic famines in China, Cambodia, and Ethiopia in the 1960s and 1970s. Reports that are strangely absent in today's media coverage. The World Vision 40-hour famine to raise money for children living in poverty overseas was kind of a rite of passage for my schoolmates and I. We wouldn't eat for 40 hours to raise money, and perhaps it was the first time that any of those fortunate children had ever experienced anything close to hunger. It horrifies me that even today, about 9% of the world's population, 690 million people, went hungry in 2019. Every year, around 9 million people, 25,000 a day, actually die of hunger. And the UN recently warned that the number could double amid the COVID-19 pandemic. The concept of pursuing a career in agricultural science, where I might actually work to make a difference in agriculture, was a very appealing career choice, although perhaps an uncommon one for a city-raised, bookish girl with a passion for horses and science. Following my Bachelor of Agricultural Science, where I focused, not surprisingly, on animal science, I pursued a PhD in animal genetics at the University of California in Davis, where I've been ever since. I love genetics. Just look at the amazing genetic diversity amongst our dog breeds, all enabled by conventional selection from their common ancestor, the wolf. If not for this genetic selection, would you seriously leave your children at home alone with a wolf? I have come to appreciate the important impact that genetics has on the environmental footprint and sustainability of our food production systems. Let's just say if you're concerned about the environmental footprint of agricultural production systems, plant and animal breeders are your natural allies. For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting on naturally occurring DNA sequence variations that result in variations and differences amongst individuals, like the colour of a cow's coat. Animal breeders are really just glorified matchmakers, effectively sophisticated Match.com, if you will. We decide who gets to be the parents of the next generation, based on sophisticated analyses of how optimally suited animals are to a particular selection objective and environment. Typically, this is based on an index that kind of weights the optimum blend of health, fertility, well-being, and productivity for that particular product: milk, meat, eggs. Like dogs selected from wolves, cattle were originally derived from aurochs. Using conventional breeding programs, achieving desired changes can take decades, especially in long-lived large livestock species like cattle, where they have a generation interval of about 3 years. That is the time from being born to having their own offspring. Innovations in biotechnology are really poised to help breeders improve the rate of genetic gain. My laboratory at UC Davis is exploring how genome editing tools could allow breeders to precisely introduce useful genetic variation into livestock breeding programs. And although CRISPR-Cas9 is all the rage these days, talons and zinc finger nucleases have been around longer, and it's perhaps ironic that almost 20 years ago I was involved in an effort to increase the vitamin E level in soy using genome editing. You might even say I was working on a project to improve the nutritional profile of plant-based protein before it was even cool. Genomes are made up of DNA, containing the collection of genes, the unique instructions detailing every characteristic of an individual, what they look like, the amount of milk they produce, resistance to disease. And genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system is made up of kind of two components, the Cas9 protein, which cuts the DNA, and a guide RNA molecule that tells the Cas9 where to cut. Bound together, they form a complex that introduces a cut or a double-stranded break in the DNA. And that break is then mended by the cell's natural DNA repair machinery. The break can either be immediately joined back together, sometimes a little bit imperfectly, so that you maybe lose a nucleotide, gain a nucleotide, which can basically throw the sequence off such that you knock out or disrupt the function of a gene. This can be used to inactivate genes, perhaps undesirable genes that do things like make an animal or a plant susceptible to disease. Alternatively, the repair machinery can be provided with a template to introduce useful DNA sequences which can replace a faulty gene with a healthy one, or change a gene to make it behave differently. In livestock, genome editing has been used to knock out a gene that makes pigs susceptible to a particular respiratory virus. I think we're all pretty familiar with respiratory viruses at this particular point in time. And it's being used to try to produce cows that are resistant to tuberculosis, a disease that affects both cows and people and which has been spreading in the UK due to transmission through badgers. It's no surprise to me that disease resistance is one of the first targets for genome editors. We lose 20% of all animal production to disease globally. Imagine if that was no longer the case. Less animals would be needed to produce the same amount of product, not to mention that animals wouldn't get sick, nor need to be treated for illness. A triple win for sustainability. And Minnesota-based company Recombinetics has produced dairy cows that are genetically hornless by introducing a naturally occurring sequence from the hornless Angus breed into dairy cattle using genome editing. And finally, my lab and others are working to produce single-gender offspring for industries where one gender is preferred as the product producer. For example, egg-laying chickens and milk production, the female produces the product, whereas in other industries, the male may be preferred. We've just produced a bull named Cosmo, shown here, that's genome edited and is predicted to produce 75% male calves. And in the plant world, there are efforts to produce disease-resistant plants in all sorts of species. This is an image of powdery mildew in wheat. Disease resistance is again a big target for plant breeding programs too. Many of these applications in this table are trying to silence or knock out a susceptibility gene. I would argue that it's better to control disease with genetics rather than chemicals. And excitingly, we can see a huge diversity of species being worked on and also the types of organizations that are working in genome editing. It really has kind of democratized the use of this type of technology in plant and animal breeding programs. Editors are developing plants with improved nutritional profile, increased omega-3 flax, designer fruits and vegetables, low gluten wheat, soy with fewer saturated fats and zero trans fats, and plants that are more resilient to a changing environment. Genome editing really just complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems that are faced by agriculture globally. However, I'm concerned that we might not be able to use this method in agricultural production systems. Why? Because for the past 20 years, I have been a participant, a weary participant, in the toxic discourse over genetically engineered organisms, or GMOs. And I've seen firsthand how emotion and misinformation, often spread by competing business interests, have been able to essentially convince the general public that this technology produces unsafe food. The disconnect between where the public is on this topic and scientists regarding genetically modified food safety is even greater than the agreement gap between whether childhood vaccines like MMR should be required between the public and scientists. It's over 50%. That's amazing. We live in a very troubled time for evidence-based decision making. I don't think I need to probably emphasize that at this particular juncture in history. I guess the fact that I'm speaking at a virtual event, taping this in my bedroom because of a global pandemic, makes that point perhaps self-evident. I fear that genome editing will become a casualty of the war on science. It will get conflated with the GMO debate, and marketers will seize on an opportunity to demonize this technology to provide a health halo over their own absence-labeled products. The Non-GMO Project has already come out saying they will not allow genome-edited products into their supply chain. On their website, it states, GMOs are now being created with newer genetic engineering techniques, some of which do not involve transgenic technologies. The Non-GMO Project is committed to preventing these new GMOs from entering the non-GMO supply chain. But will there be a discussion of the resultant trade-offs? The disease burden that could have been addressed? Because diseases are still going to infect plants and animals, and absence labeling doesn't solve that problem. Genome editing could, but will it be allowed to do so? Disease resistance seems to me to be a trait that should be embraced by sustainability advocates rather than eschewed. What message will prevail in the communication around genome editing? The promise or the peril? I hope it's the former, because the promise is too important to agricultural sustainability to forego. And so while it is true that I do support the use of new breeding techniques and their purported advantages, as this Twitter troll points out here, and I guess I do kind of have to own that I have had kind of the same hairstyle for the past 30 years, although I'm not 100% sure I understand how one relates to the other, but I guess if you're a female in psychom, that's just part of the baggage you get along with it. But for over 30 years, I have had a passion for genetic improvement of our food-producing species, and I've spent my entire career working in that field. I see huge potential for genome editing to make CRISPR cows and Talon tofu. And you know what? Do you know who else didn't change their hairstyle very much? This guy. Well, I hope you enjoyed our TED Talk. I was a little bit challenged when I was tasked with doing a TED Talk from my bedroom in the middle of a pandemic, but fortunately we have recently gone from being empty nesters to being very full housers because the kids are back from college because of the pandemic. And so fortunately, one of my sons, Alec, is very good at Adobe Premiere Pro. And so he is the one that has made all the great transitions and made this look a lot more professional than it would have if it had just been up to mum. And so I'm going to bring Alec into screen. You can see the whole thing here has been done with a green sheet, and used to be my bedroom lamp. It's now turned into a studio lamp, and it's actually been a bit of fun to put it together, but I really thank you for your help in that regard. And this generation are the ones that are going to inherit this society that doesn't respect evidence, and so I think it's such an important thing to teach our kids to think critically and to base their decisions on evidence. And that's why you're wearing your mask, because science says that's the smart thing to do in the middle of a pandemic. And there's really no greater gift we can give to our kids than a society that bases decisions on evidence. I hope that they are able to counter misinformation and understand that they need to look to what the consensus is of the scientific societies of the world, because otherwise we have major problems that come around if ideology and politics drives basically what should be evidence-based decisions. And so I'm going to finish with a little musical parody that my lab put together, my grad students. We're all about SciComm. And so my PhD student, Joseph Owen, put together a musical video that displays how enthusiastic they are about genome editing. And it's called Can't Stop the Feeding. Joey's also actually the student that produced Cosmo, the genome edited bull. So he's both a very good genome editor and also a good video editor as well.
Yay, Alison, that's fantastic. Yeah. Man, thanks for your passion. Thanks for sticking it out for 30 years. I know you're out there putting yourself out. You're getting trolled. God, you're really brave and you're fun and you're funny. And I have some questions, but Adam, I'm going to hand it over to you to start with the answering of the questions, and then I'll save mine when it's right to ask.
Absolutely. Thank you so much again for presenting here as well. Thank you, Dr. Van Eenennaam. Yeah, I guess let's get started with some questions that people have. Just again, if you have any, for anyone who's watching this, if you have any questions, there's the Q&A button at the bottom of your screen. You can click that and then you can ask questions there. You can also click there to see what other people are asking and upvote questions that you would like to see answered. The first question we have is from an anonymous attendee. So Dr. Van Eenennaam, how much opposition to GMOs are there in countries where people are experiencing famines?
That's an interesting question. Thanks. So I guess the answer to that would be that I've traveled quite a bit through Asia and Africa, which is where a lot of food insecurity is, and they face similar problems to what scientists face here. So the scientists are supportive of the technology and have developed amazing disease-resistant applications like the banana wilt resistance banana in Uganda, for example, even golden rice. So there's great support from the scientists, and then there's very active opposition, often funded by wealthy European NGOs that are down there fermenting fear around this technology. And some of the stories you see are, they kind of go after the fears of the population. So in Africa, one of the common pieces of misinformation is that this technology, if you eat this food, it'll be associated with infertility in some way. And they really are just almost criminal in the misinformation that they're putting out there. And so the general public, as has happened here, hears all of this negative information, and the voice of scientists saying, actually, that's not what the data says, get drowned out. And so I think that at the moment, particularly in Africa, that there could have been some tremendous benefit from having insect protected crops, and because that's leading to, there's a number of crops at the moment that are getting basically ravaged in the insect outbreaks, and the technology exists, but it hasn't been able to be used there. And I think, to me, I guess I find it kind of criminal that well-fed countries get to dictate to developing countries what technology their scientists are allowed to use to genetically improve their own crops. So I think that in my experience down there, and obviously I usually talk to the scientists, they're supportive, but it's political interference and misinformation by basically quite often groups from the global north.
And thank you. And quickly for some questions that Dr. Challa was able to answer earlier. People were asking, where do we find information on his workshops, and he has posted that answer directly if you go under the answered Q&A questions. In the meantime, we also have another question to Dr. Van Eenennaam, which is, you make some persuasive arguments for GMOs. What are your thoughts on the boundaries of using gene editing in human applications?
So I'm an agricultural scientist, so I usually talk about food applications. It's kind of ironic to me because when it comes to genetic engineering, we use that routinely in medical applications. For example, insulin is produced using recombinant DNA technology, and we use it in food processing to separate curds and whey when we're making cheese. But when it comes to genetic engineering and agricultural production systems, that's when there's kind of this bright line drawn in the sand. And I think that when it comes to medical applications, that the beneficiary of that edit, and if we're talking here about trying to help a person that's got maybe a genetic disease, I think it's a lot more obvious to that individual the benefit, and potentially a lot more severe the outcome if they refuse the technology. So if I've got a fatal disease and I have a potential to be cured of that disease, that's kind of my decision and my benefit, and it doesn't, other people, that they can make their own choices about whether they want to be edited or not. And I think that's what you see is there's a lot more support for obvious beneficiaries when the person involved has choice over it. And I think what the SciComm literature shows is people don't like feeling they have no choice in a technology. And so I think that's where some of the pushback on the GMOs is, well, I don't know, you know, I have no choice in that technology, and maybe the benefits aren't as obvious to the general population because they've never dealt with insects or plants that, you know, are all dying. And so it's not as obvious, although I would argue it's had tremendous social benefits in terms of reduced insecticide use, for example. But to an urban person, it's maybe not as obvious what the benefits are.
Right. I guess along these questions, we have a question from Ankit Sabharwal, and this is to both speakers. So as a part of your outreach, are you involved in explaining the benefits of GM plants and animals, and I guess the CRISPR technology in general, to the general public and young students? And what are some of the challenges that you've seen explaining it to the public and young students?
Why don't you go ahead, Anil?
Sorry, I didn't catch the question. Sorry. Can you, Adam, could you
what are some of the challenges involved in explaining CRISPR or other genetic engineering technologies to, in your case, students?
I think, to me at least, the fun part and the easy part would be to use a lot of analogies. And so, for example, I use the analogy of a programmed biomolecular nano drone for CRISPR, right? So I think once they capture their imagination, I think it's a lot easier to explain. And I always take the side of technology as a tool, in the sense, you know, it's like fire. Can you burn things, or can you cook food, kind of thing, right? So I think I encourage a very healthy debate. And to be honest, what I feel is when I let the ground to be neutral, I notice that people are a lot, I mean, at least students are a lot more open. I do see that the debate, I see that, oh, this is bad, this is good. But I say, as humans, we don't always deal with good and bad, black and white, but what's optimal. And okay, to quickly answer your question, I don't see a lot of resistance, at least in students. I hope I answered the question.
Yeah. Thank you.
And I guess I'll just say, I think I always like to frame it in terms of what's the problem we're trying to solve, rather than focusing on the technology per se, because if it's a compelling problem, then that genetic solution, I know I'm a geneticist, so I have my own confirmation bias, but that's a better solution than having sick plants and animals or sick humans. And so that's kind of where I tend to go with that. And I see Perry asked a question about this particular state and its opposition to GMOs, but I think it's interesting that, you know, there's some new products that have come on the market in the last couple of years. For example, the Impossible Burger that includes genetically engineered heme, and that has had market acceptance with no problem. And so I don't really buy the narrative that all the general public is opposed to GMOs or won't purchase it. I think if the product is what they want and they, you know, they're buying it because it has some purpose in their mind or it solves some problem in their mind, then it's not the technology per se that's stopping them from buying those products. And I guess I just would love to let the people have access to the technology and actually see if they do buy it. I think, you know, Europe kind of made the decision not to let it on the market, at least as it relates to human food, although of course they feed their animals GMOs, but that really removed that choice from that consumers. And so I'd love to see some more gene edited products on the market to let the consumers make those choices in the marketplace.
And I guess bouncing around, Dr. Challa, do you think your classes for students and then by extension high school teachers are also influencing the students' views on GMOs?
Just one point is the workshops that we do is for undergraduate educators, but I have very little interaction with high school teachers as yet. But we have really fun debates about GMOs, and especially in the CRISPR realm. So it does, I think, first of all, I think it's already, it's out there, people are talking, at least in my classrooms, almost all students. I've seen the difference between 2015 and now. People didn't hear about CRISPR in 2015 so much, but now all freshmen come in and say, I mean, they have an opinion one way or the other. So it's actually nice to, we do these sessions where students are given different scenarios and they have to discuss as teams. And I kind of force them to think from different angles as a sociologist or an ethicist or a lawyer or a business person and as a scientist. And so you talk about this, and I agree with Alison about the problem statement, right? So I think we put a scenario out there and say, okay, what do you do as humans, right? And it's not about this or that, but how do you bring all these perspectives together to solve a human condition or a bio scenario situation.
I'll ask the next question, Anil, so stick around.
Mm-hmm.
So do the CURE, do you call it the cures?
Cures.
Cost more to run than a typical intro biology course?
I don't think so. For example, I've been trying to do the budgeting myself, and I think if we are clever and if we define the scope of these cures, they don't have to be, or they don't tend to be much more expensive. If you especially don't count in the instrument costs and the capital costs, it's not necessarily different. In fact, I think cures are more conceptual than actually the physical aspect of it. So I think we can be creative in organizing cures in an economical way.
Okay, and another sort of a two-part to that is it kind of tricky to get the administration to buy in on it?
Yes, I had difficulty. I mean, I had the initial things were challenging, but once they saw student response, it was an easier sell. I'm not saying it's easy, but it's an easier sell because students are really, really excited, especially now that we have a peer-reviewed research article published by students. That is a big, I think all schools would love to see their undergrads published, not necessarily because they are actually getting students to engage with authentic work. So I think it is slowly. I mean, I have to be fair. It's not just me. Nationwide, cures have gotten a lot more, and on other authentic research experiences have gotten a lot more positive attention. So things have actually improved over the last few years that I have seen. Yep. So administrators are taking up this in a very positive way.
Yeah. So there's a couple of other questions. Adam, I'm going to kind of keep rolling here, if you don't mind. So a question is asked that was something I had in my mind too. Let's see. Does a course focus, I just found it, on plant editing, animal editing, or human editing? I'm wondering if the students get to look at their own DNA.
Not in my course, not in my course, but there's a really nice course at University of Minnesota where I should know. I mean, I know him very well, but he does the 23andMe kind of stuff with students. He's got some really good stories. In my class, it's actually students who are really focused on kind of research or medical professions. So they're freshmen. You know how freshmen are. I mean, it's very, I like to work with them, but their perspectives are still a little green and they are developing. So that's why I actually enjoy working with them. But I don't necessarily focus, I mean, it's heavily focused on zebrafish with a human disease leaning towards it.
And Dr. Van Eenennaam, we have a question for you, which is, I often encounter, reading off here, is I often encounter the argument that agriculture is, quote, natural, and what's natural is better. Particularly that there's a question on whether we can be 100% sure that a modified crop would not have environmental and detrimental effects. The comment goes on to say that, to ask, is there a bit of a disconnect with that it's not responsible to say that we can be 100% sure of anything, and what is important to the public?
Yeah, I always am intrigued by the idea that natural is better, and I guess COVID's natural. Are we enjoying that? You know, botulism's natural, the plague is natural, all diseases are natural. So I guess I don't really think that's necessarily a beneficial thing. I think it can be good and it can be bad. Depends what the thing is. It doesn't matter. But the argument that agriculture is natural, I guess I can't really buy into that either. I think that probably agriculture is the system that has the biggest environmental impact in our world, and the science involved in growing our food is, I mean, you know, oryx are not what we raise. We raise animals that are incredibly different to their ancestors, and similarly, I think if you look at corn relative to its ancestor, Teosante, you know, we have dramatically changed things through human selection, for better or worse. I mean, I guess if it's for worse, then we need to figure out which 4 billion people are no longer going to be on the planet because we wouldn't have enough product to feed them. So I think that, in fact, agriculture does use a lot of things that have had human intervention, and that's not necessarily a bad thing. I think in medicine, we surely hope that there's human innovation there to develop better drugs and cures for diseases. So why would we want the industry that has the biggest environmental footprint on Earth to only use natural methods? I think that that's a recipe for disaster. But I think you mentioned 100% certainty. There's no 100% certainty in anything. You know, I'm pretty sure the sun will come up tomorrow, but I, as a scientist, cannot guarantee that. And I think what I often see in the field, especially as it relates to the risk of GMO crops, is that there's a request to prove safety in the future. And no scientist can prove something in the future. We can take the evidence we have from 30 years of growing these crops now, and the overwhelming scientific consensus of every major society in the world, and let that drive our decision making. But I cannot prove future safety of anything. And I think that what has been seen is that from the products that have come to market, there's been a tremendous impact in terms of improved management for farmers, decreased insecticide use in the case of BT, and that's helped to increase the income, especially the biggest 90% of the people using genetically engineered crops are in the developing world, small scale farmers in India and China that are growing insect protected cotton. That's a really big proportion of the farmers that are growing. I think it's 18 million farmers globally. But we very rarely even hear that discussed. And so the narrative is out of whack with where the benefits have actually been obtained. And it's stopped a whole bunch of really useful applications from basically being utilized by farmers globally. And I think that's what frustrates me is that there's no data that says this is an unsafe technology. We've looked at the risks and the benefits outweigh the risks, and yet we can't use this technology, at least genetic engineering. And I fear that genome editing may go the same way.
Thank you, Alison. Well, we're right at the end of this session, scheduled for 9 o'clock Central Daylight Time. There's a couple of housekeeping, rule things to take us out. But before we do, Dr. Challa, do you have a couple of last comments, and Alison too? And then Adam will mention a couple of things just to keep in mind before we say goodbye.
So I'd like to echo what Alison just mentioned. I think the science is exciting. The science is very open, but I think it becomes a human and a political issue more than a scientific thing. I think especially, I remember one time I was invited to a panel. They wanted me to be the scientist on the panel to talk about GMOs. But the moment I said that, they wanted me to almost say that GMOs are bad. And I said, I'm not willing to say that. And so I was actually disinvited or whatever. I mean, I was asked to leave because I said, look, I'm open to your points of view, but I'd like you to be open to my points of view. And I'm going to bring the scientific aspects onto the table. And they already wanted me to have a biased agenda. And I said, I can't do that. So it's almost like people are not willing, some people are not willing to look at the science. And they, at least in social circles, they seem to have a larger or a louder voice. And I think we should continue to do something, I mean, like what Alison is doing. I think we just have to keep talking about the science and focus on the strength of the science and what we can do with it to make more and more people aware of it. So, thanks.
Yeah, I mean, I guess I'll just finish that we've been giving people a science for 30 years and we're losing miserably, not only in this debate, but also in the vaccine debate and even wearing, face masks. And so I think that the communication has to change to go to people's values. And quite frankly, that's what's frustrating is that most people I know want to decrease the environmental footprint of agriculture. They don't like to see animals get sick. They don't like the waste and the chemicals that you need to treat sick plants and animals. And so here's a technology that could potentially fit in with all of those stated values. And so it's really our job to reframe the discussion from being a nerdy scientist to being connected with the fact that I share your values. I want my kids to grow up in a society that has access to safe technology, and we need to get this next generation. I like this next generation. I teach them, and they've got it. They've figured out some of this misinformation, and I'm hopeful that actually this whole pandemic and the whole science kind of chaos that we have at the moment, that it'll make them more critical thinkers and more skeptical of information they're being given that isn't backed up by evidence and science. So I'm hopeful for the future because I think this generation, this pandemic maybe will be an influencing force on their life to make them appreciate how important science and evidence is to make their decisions.
Thank you so much. Keep at it. We need you. What you're doing is so important. The education, teaching the teachers and teaching the students. Just a wonderful discussion. So I think we'll thank you again right after. Adam, can you walk us through a couple of comments for all of the listeners out there?
Absolutely. Thank you everyone for attending. Thank you to Dr. Van Eenennaam and Dr. Challa for logging on and being willing to answer questions. For those people who are registered for the full conference, they'll be available at a breakout session that's linked on Canvas, and I'll be sharing that shortly. For everyone else, thank you so much for coming. Tomorrow morning, this conference will continue focusing on COVID innovations starting at 8:30 in the morning, Central Time. And in the meantime, please check out the GenomeWritersGuild.org, our website, for any upcoming events or to join the group. And then follow us on Twitter and Facebook at Gene Writers with an Gene_Writers. And again, thank you everyone for coming on. It's been really a pleasure. I'll be posting these questions further down.